Hey everybody, let's go over the quiz answers for our lecture on molecular motors and cargo transport. All right, so here's the pre-quiz. Question number one, increasing temperature. So we were talking about diffusion and the need for cargo transport. So this is a question about diffusion. Increasing temperature will increase the rate of diffusion. All right, that's if you increase the temperature, things will move around faster, and generally things will diffuse faster as well. All right, molecular motors can convert chemical energy into motion. Motility and gliding assays for motor function are performed in vitro. Remember, this is the assay where we attach the motor protein to the glass slide, and then it pulls the filaments around, and you can watch them gliding around under a microscope. All right. Dynein and kinesin motors transport cargo along microtubules, and HSV1 can be used as a neuronal tracing reagent. And the answer to that is true, especially if we genetically modify it and add GFP. All right. Post-quiz now, post-discussion quiz. For in vitro motility assays and gliding assays, which component moves across the microscope slide? That's the cytoskeletal filaments. All right. What direction is HSV1 transported in the nerves that lead to the trigeminal ganglion? So we talked in class about the pathogenesis of HSV, herpes simplex virus. And it's important to know that anterograde is going away from the center of the cell and retrograde is going to the center of the cell. So we can think about which is happening there. But it's it's actually, if you remember all the steps and the different things that happen during herpes infection, it's a little bit simpler to answer this question because it goes in both directions. So the answer here is anterograde and retrograde. Most of you had retrograde, which was correct for the first stage of infection, but then remember it comes down in the anterograde direction during the the flare-ups as well. All right, next question. Kinesin motor proteins bind different cargo through their different tail domains, and dynein motor proteins bind different cargo through different adapter proteins. Remember, there's lots of different kinesins, all with different cargo binding domains, and there's essentially just one dynein cargo transport protein with lots of different adapters to let it carry different cargoes in the cell. All right, the thermal ratchet mechanism. Remember, this is how this is the mechanism that allows cells to use actin polymerization to move forward for cell motility. So this relies on B and C, cytoskeletal filament polymerization, specifically actin polymerization, and then the Brownian motion. You need to have the plasma membrane motion is what creates the space for the filament to polymerize and then keep on pushing forward. All right, and the last question is, which of the following can be used for neuronal tracing? Viruses, we've already gone over that. Proteins, so there are certain proteins that if you inject them close to neurons, they'll be transported in the anterograde or retrograde direction. Proteins, actually there's a lectin protein, wheat germ agglutinin, that's used for this, and there's other proteins as well. We can also use electron microscopy. This was in the table in the slide. We could just take lots of sections of electron microscopy and recreate a three-dimensional, very high-resolution image of tissue with neurons in it, and we can use that for neuronal tracing. But we can't use Western blotting. Western blotting, to my knowledge, has never been used for neuronal tracing. You lose all spatial resolution. You have to take a tissue and lyse it, crush it, lyse it, squish it all together in order to run it on a gel. And when you run it on a gel, you lose all the spatial information. So you can't do neuronal tracing with Western blotting. All right, that's the end of the video for this lecture. Good luck studying for the exam, everybody, and I'll see you in the next lecture.